In this lesson, we are going to begin talking about cells. Cells are the basic unit of structure and function in all living things, so they are crucial to our understanding of biology in general. And I'm including here a picture of some of the earliest drawings of known cells. These were drawn by a man named Robert Hooke, who was one of the first to use microscopes. And these are a drawing of cork cells. So this was some of our earliest understandings of the fact that all living things are made of cells. And in this lesson, we're going to just focus on an introduction to cells at this time. Now, as I said, cells are the smallest unit of life, the smallest piece that we can call a living thing. They are organized, highly organized. They can reproduce on their own, and they can live on their own in some cases. Here's an example of an animal cell. This would be from a multicellular organism. This cell could not live on its own. It needs the entire body to survive. But this cell is a single-celled organism known as amoeba, and amoeba can live on their own. Just a single cell is their whole. The whole organism is just one cell. There were many people involved besides Robert Hooke in our understanding of cells. And in particular, there were three individuals who developed what we now know as the cell theory. And the cell theory states that cells are the most fundamental part of life, and that all cells have to come from pre-existing cells. Now, this was a new idea when it was first developed, because at one time people thought that, in fact, cells, living things, could come from non-living matter. But we now, of course, understand that all cells have to come from other cells. The three individuals that were involved in the development of this theory were Schleiden, Schwann, and Virchow. So we have, oh, I apologize. We have Schleiden. Schleiden studied plant cells, and he first discovered that all plants were made of cells. Then we have Schwann, who studied animal cells and discovered that, in fact, animals also were always made of cells. And then finally we have Virchow, and Virchow contributed that last piece, this idea that cells can only come from existing cells, and together this makes up the cell theory. Now, cells will differ in size, shape, and because of their differing in size and shape, they differ in function. But they do share common structures. Something you'll see in all cells is a plasma membrane, and we're going to spend a good deal of time talking about what it's doing. All cells, actually not all cells have a nucleus. That's not correct. All cells have DNA. Let's be more specific. And all cells have something we call the cytoplasm. Now, beyond that, there are a lot of differences between cells, but these things are in common to all cells. There are two main types of cells. The first type we call prokaryotic. Pro means before, and the karyo is the nucleus. So these are cells that evolved before any cells had a nucleus. So these cells have no nucleus, and in fact they have no organelles or small parts inside that have a membrane around them. They have no membrane-bound organelles. What we're talking about here are bacteria. Two groups of bacteria, archaebacteria and eubacteria, and these are some examples of them. But these cells have no nucleus, no membrane-bound organelles of any kind. Very simple cells, very small cells. The second type of cell is a eukaryotic cell, and EU means true. So these cells have a true nucleus. They do have a nucleus, and they have other membrane-bound organelles. They tend to be larger in size and much more complex. Examples of these: plant cells, animal cells, fungal cells, and then those that we call protista that we'll learn about later. So here's a variety of cells. Here are some plant cells. Here are some animal cells. This is a generalized diagram of an animal cell. And you can see inside it's much more complex than that prokaryotic cell you looked at a minute ago. Now we can compare prokaryotic and eukaryotic cells. The DNA in a eukaryotic cell is found in the chromosomes. In prokaryotic cell, it's not. In a prokaryotic cell, there's just a region called the nucleoid region where you'll find the DNA. In a eukaryotic cell, you'll find it in the nucleus surrounded by a membrane. So it's behind a membrane, it's inside a membrane. They both have a cell membrane. They both have cytosol or cytoplasm. Eukaryotic cells have larger ribosomes in the cytoplasm. Eukaryotic cells have many membrane-bound organelles. Prokaryotic cells have none. Prokaryotic cells have a cell wall and capsule. Eukaryotic cells have a cell, some of them have a cell wall, none of them have a capsule. And of course, I do want you to notice the size difference. Eukaryotic cells are much larger. No matter the cell, though, cells are all small. Even the eukaryotic cells, that's still very, very, very small compared with our general reality, and that's why we didn't even know about cells for so long. The question is, why are they so small? And this has to do with some mathematics, actually. As a cell grows larger, its volume will increase faster than surface area. The volume is cubed while surface area is squared. So surface area increases much more slowly. The volume, think about the volume of a cell, is where all the metabolic activity takes place. The surface area is the area for diffusion of substances into and out of the cell. So you have to think about all the activities going on inside the cell and the fact that exchange of materials has to happen at the surface area. By the way, this is the largest cell that I think we know of, one of the largest. This is actually a single cell, and it is an egg cell, a human, I think it's a human, egg cell, which is actually visible to the naked eye. One of the largest cells, but it's about the size of maybe a little mark of pencil on a paper, like a period on a piece of paper. But it's the largest. Most cells are much smaller, too small to be seen with the eyes. So let's take a look at this. As a cell grows larger, it has to work harder to get nutrients and to get rid of wastes because of that change in the surface area compared with the volume. So we look at something called surface area to volume ratio, or SAV ratio. If the SAV ratio gets too small, the cell will die. So if we look at a cell like this, which is a large cell, 20 microns, with it's just a single cell. We see that it's hard to see this, I know, but the surface area to volume ratio here is 0.3 for that cell. The next cell, if we break it up into 8 cells instead of just 1, we increase the surface area. Now the surface area to volume ratio is 0.6. And finally, if we break that up into cells that are only 2 microns instead of 20 microns, the same volume, we now have a much greater surface area, and our surface area to volume ratio here is 3.0. So you see it's much better for the cell to have more surface area. It can more easily move substances in and out. Now some cells can be larger because they solve the problem of surface area by either growing long and thin or having a folded surface. An example here, this is cells of the intestine, and you can see here along the top how it's all folded up. All of that is increased surface area so this cell can get rid of waste and gain nutrients more easily. Another example here, this is a nerve cell, a neuron, which is a nerve cell, and neurons can be quite large. They can be there's one neuron that connects your big toe with your spine. That's a very big cell, but it's very, very thin. It has this long part called an axon, and because of having that part, that long thin part, it can grow quite large and still have enough surface area to get rid of waste and to gain nutrients. Now later on we'll talk more specifically about eukaryotic cells, but for today, focusing just on some characteristics of prokaryotic cells. As I already said, the DNA is found in a nucleoid region. It does not have a nucleus. It does not have a membrane around it. And prokaryotic cells have a semi-rigid cell wall containing a capsule, sticky polysaccharides around the surface, and something called pili, which makes it look kind of furry. And so because of this, these bacterial cells basically can stick to surfaces. Much more so than a eukaryotic cell. This is very important because many of them are pathogenic or at least parasitic, and they need to live in or on other organisms, and so this allows them to stick to those other organisms. The capsule is protective. The polysaccharides that cover its surface and the pili that cover its surface allow it to stick. Okay, I'm going to cut this one short, and that's it for this lesson, and we'll talk more about eukaryotic cells next time.